everyone, and welcome to today's webinar, Ultraplex Multiplex Technology, Tagging Technology for Mouse and Human Multiplexing. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Leica Biosystems. To learn more, please visit leicabiosystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. With that, I'd like to now welcome our speaker, Dr. Helen Snyder, Director, Multiplexing Product Management at Cell IDX, a Leica Biosystems company. Helen, you may now begin your presentation.
Thank you, Kaylee, for that introduction. Well, I am delighted to be here. I really appreciate the opportunity to talk to you about Ultraplex, which is our tagging technology for generating both mouse and human multiplex panels with both fluorescent and chromogenic readouts. So what is the Ultraplex technology? Well, it is a platform technology which on its first iteration, we've applied to multiplex IHC. However, it has the potential to be applied to many different readouts. Our primary goal was to develop a technology which was extremely simple, rapid, and easy to adopt. Something you could run on equipment you currently had in your laboratory. And as I said, we also wanted it to be rapid so that you could stain in the morning, scan after lunch, and then analyze your data the same day. So how does it work? The basis of the technology involves labeling individual primary antibodies with multiple copies of a specific peptide-like tag. Each primary antibody is labeled with a different tag, which is what allows us to add all the antibodies simultaneously as a cocktail. So using fluorescent multiplex IHC as an example, and I apologize, I don't have a pointer on this presentation. So if you could look at the far left image, we'll just start from there. So the tissue undergoes a single epitope retrieval step, followed by the application of a cocktail of primary antibodies, each labeled with a different peptide-like tag. The tissue is incubated, washed, and then a cocktail of anti-tag secondaries, each labeled with a different fluorescent tag, is applied to the tissue. This means that you get amplification from the multiple copies of a specific tag on the primary antibody, and also from multiple copies of a fluorophore on the specific anti-tag secondary. So sometimes people attempt to set up multiplex IHC using primary antibodies of different species. So this is rather limited in that you need good antibodies of different species. You might try to use one mouse antibody and one rabbit antibody, and then detect with, for example, anti-mouse and anti-rabbit secondaries. However, sometimes this is impossible to do, as good antibodies aren't always available of the specificity you want in a different species. And then what if you want to use all rabbit monoclonals, which actually even all the same isotype? So what the Ultraplex tagging does in effect is it makes each antibody its own species, as each primer is recognized by the tag that we add, and then the anti-tag secondary. So this gives us huge flexibility. It doesn't matter to us what species the antibody is. We can use all of the same species in the same Ultraplex cocktail. So as I said, our primary goal was to develop something that was simple and rapid. And this is very simple and rapid. In fact, it uses simple standard primary antibody, secondary antibody workflow, except instead of using one antibody at a time, it uses cocktails of antibodies. So our second goal was to develop a multiplex IHC technology with both fluorescent and chromogenic readouts, which would take you all the way from your preclinical biomarker studies in mouse all the way through to your human translational studies using our research use only reagents. And of course, ultimately to clinical, but obviously we're not quite there yet. So if you look over to the left, these are our human translational solutions. The first two panels are fluorescent and the lower two panels chromogenic. Over there on the right, the upper two panels are our mouse preclinical tissue biomarker solutions. And below that, we have really our solutions for multiplexing of custom biomarkers. So for a little more detail, if you go back up to the top left, these are our human ultraplex fluorescent panels, which are ready to use. So this is HI1 through 6. They are predefined 4-plex panels, which if you're working in the area of immuno-oncology will make a lot of sense. They include different lineage markers for immune cells, as well as checkpoint markers and tumor markers. Below this, we have ultraplex human fluorescent modular panels on demand. So modular is an approach which allows you to look at a menu of qualified biomarkers and choose the 4 that you want to incorporate into your panel. You choose the fluorophore sets and you place the order. Below that are our chromogenic human solutions, ultraplex human chromogenic panels ready to use are CPO1 through CPO3, and I'll show those to you in a minute. Below that are our chromogenic panels on request. So basically, if we have qualified antibodies by fluorescence multiplex IHC, then we have a really good chance of being able to apply those to chromogenic multiplex IHC. So if you see biomarkers that you like, just ask us about chromogenic panels. Then over there to the right, the mouse ultraplex fluorescent modular panels. With mouse, we went straight to modular because people always have different panels that they like to use. So again, it's the same approach. You take a look at the menu of qualified biomarkers, choose the 4 you want, the fluorophore sets, and then place the order. Again, if we have qualified antibodies by fluorescence, then there's a good chance that they will work in chromogenic multiplexing. So you can place a request for a mouse chromogenic multiplex panel. Below that, we have UltraTag and PathIDX. So UltraTag are our do-it-yourself multiplex labeling kits. So you are able to label your own antibodies. Each tag labels 1 antibody. So if you want to create a 4-plex fluorescent panel, you would choose four kits. We also have kits to create chromogenic multiplex panels too. And if you would rather that we develop your custom panel, we can do that through PathIDX. So ultraplex fluorescent and chromogenic multiplex IHC, how does it work? So again, if you look at the top of the flow diagram, we have UltraTag labeling. So this is what we do at CellIDX in manufacturing, but it could also be what you do with the UltraTag kits labeling your own antibodies. This is followed by a single antigen retrieval step, and then you apply the cocktail of primary antibodies simultaneously on the tissue section, whether you then proceed to fluorescent multiplex IHC or chromogenic multiplex IHC. So looking over to the left to begin with the fluorescent multiplexing, after you've added the primary antibodies, you incubate, wash, and then you come back with a cocktail of anti-tag secondaries, each labeled with a different fluor. And the runtime for this is very rapid. On your BondRx, it will be about 3.5 hours. The image that I'm showing below that is a staining of breast cancer. So the ring membrane staining in red is the HER2 molecule. The blue nuclear staining is the estrogen receptor, and the green nuclear staining is progesterone receptor. Where those are two are co-expressed in the nucleus, it appears as that kind of turquoise color. And then the pink nuclear staining is Ki67. So with this one panel on one tissue section, you can actually subdivide breast cancer into the 4 main subtypes. If we now look at chromogenic multiplex staining on the right, you can see again you add the same cocktail of 3 primary antibodies, although the concentrations are a little different for chromogenic. You then add 1 anti-tag labeled with AP and a second anti-tag labeled with HRP. You develop with the AP and then the HRP chromogens. Then the first anti-tag HRP undergoes an HRP arrest step. So this is to kill the HRP so it does not interfere with the subsequent staining step, which is to add a second anti-tag labeled with HRP and then develop with the second HRP chromogen. So the image that I'm showing you there, and I'm sorry, it's quite small, but you can, and we've made it a little more complex because instead of red HER2 staining, it's now blue on the chromogenic staining. But you can clearly see the blue membrane staining for HER2, the red nuclear staining for the estrogen receptor, and where there's co-expression of the estrogen and progesterone receptors, the nuclei appear orange. So again, as I mentioned, it's a simple, rapid standard, uses a simple, rapid standard primary antibody, secondary antibody, IHC workflow using cocktails of antibodies. We have both human and mouse biomarker panels, and the technology is primary antibody species independent. So why is that important? Well, for us, it's important in that it gives us really ultimate flexibility in the antibodies that we use. In fact, frequently, a lot of our antibodies will be rabbit monoclonals. They're very high affinity antibodies. So we can easily make full panels using rabbit monoclonals or all mouse monoclonals. It doesn't matter to us. And so why would it matter to our customers? Well, if you're also planning on making your own multiplex panel using UltraTag, then it's nice to be able to use multiple antibodies from the same species. But also you might have a favorite antibody that you've used for a lot of different studies and you want to include it in your panel, and maybe there are the same species also within that panel. Then again, it doesn't matter for you, depending on what the primary antibody is. You can use it regardless of the species. Another advantage is that it has a single antigen retrieval step for IHC. Again, why is this important? Well, some multiplexing technologies use iterative rounds of antigen retrieval. So it's stripping and staining, stripping and staining, stripping and staining. And this can affect the levels of antigen expression at different stages and sometimes the tissue morphology. And so we don't have this problem at all with UltraPlex in that we don't have to care about the order in which the antibodies are added because they're all simultaneous. And also we don't have to worry about the expression or the morphology because there's only the single antigen retrieval step. Also because increasingly the tissue biopsies that we're looking at are increasingly small. Often we'll be looking at, you might be looking at in-treatment biopsies, and frequently these are small needle core biopsies. And so we've had customers who are working on needle core biopsies who are a little concerned in case with a small tissue sample that if there's a very prolonged procedure that they actually might lose the tissue from the sample. But because we only do a single antigen retrieval step, that's definitely an advantage of UltraPlex. So the fluorescent multiplex IHC panels we have 4 to 5 plex panels. Routinely we do 4 plex panels, but we can do 5. For chromogenic, we can do 3 or 4 plex panels. So what I'm showing on the flow diagram is obviously a 3-plex panel, but if we wanted to create that as a 4-plex panel, all we would do is we would add all the primaries simultaneously, all 4 of them at the beginning. And then after what I've shown you here, we would do another HRPRS step and come back with a third anti-tag HRP and a third HRP chromogen. UltraPlex chromogenic multiplex IHC and Leica instrumentation. Why is this special? Well, UltraPlex is a chromogenic multiplex staining technology, which is simple, fast, and fully automated. It generates beautiful images and data on biomarker expression and spatial localization of key cell subtypes using the BondRx staining system and the Imperial GT450 scanner. So the image that I'm showing, the flow diagram over there to the left, is the one I've shown you before. But I did just want to emphasize here the reagents that you would need for the staining. So obviously you would use your usual bond wash solutions. and your BondRx epitope retrieval solution. And all of the images that I've shown you so far have been with the ER2 epitope retrieval solution. You would also need to order the Bond polymer refined red detection kit and the Bond blue chromogen. But included within the Ultraplex kit are the antibody cocktails and the antibody diluent, the HRPRS solution, the yellow chromogen, and purple hematoxylin and blue hematoxylin counterstains, so that you have the option of one or the other. If you have the Bond RX 7.0 software, BXD25 or above, the Cell IDX chromogenic protocol, which is Cell IDX RBY, is preloaded onto the software. So you just need to load your reagents and hit the run button. The image over there, which I'm showing, was produced on the Aperio GT450 scanner. We routinely use the Leica Versa scanner for both chromogenic and bright field, but I really appreciate the speed of scanning using the Aperio GT450. It's very rapid. It takes seconds to minutes. So it's very applicable to high throughput staining and imaging. So what are the advantages of multiplexing? Well, over there on the left, we're showing standard IHC staining. There are 3 sections, each stained with a different biomarker. On the right, we show ultraplex staining, where one section is stained simultaneously with 3 biomarkers. So for both of these staining technologies, it's simple, rapid, and uses standard laboratory equipment. But the one over there on the left uses 3 slides and 1 biomarker per slide. So this does waste precious tissue. As I mentioned earlier, increasingly tissue samples are smaller. For example, the needle core biopsies, where often you might get 3 or 5 sections per core. And these have to be applied to different detection technologies. Maybe you might be looking at RNA expression as well. So to use 3 sections for 1 staining is wasteful. So this is solved using the Ultraplex technology. Also, you only see a single biomarker per slide, and many of the different subpopulations of cells we look at now are only identified by multiple biomarkers per slide. So you aren't able to detect those. Over there on the right, you can see 3 biomarkers shown on 1 tissue section. So again, this is tissue conserving. It frees up those other sections for different readouts. It also gives you multi-parametric analysis. So you can look at co-expression of 2 biomarkers on the same cell population. And it also gives you information on context. So for example, the tumor cells are shown there in yellow. CD4 cells are shown in blue and CD8 cells in red. So you can see where the immune cells are relative to the tumor and also relative to each other. So this is showing our Ultraplex Human Chromogenic Multiplex IHC ready-to-use panels. The first panel, CP01, targets CD8, PD-L1, and pan-CK using red, blue, and yellow chromogens. CP02 targets CD8, CD4, and pan-CK, again using red, blue, and yellow chromogens. And CP03 stains for CD68 and CD163, and also for PD-L1. So this is basically looking at PD-L1 expression on different subsets of macrophages. The chromogens we use are the red Bond polymer refined red detection kit, for blue, the Leica blue chromogen, and for yellow, the cell palette yellow chromogen, which is included in the CP01 through CP03 kits. So this next slide shows our CP01 through CP02 and CP03 staining profiles. If you look over there to the left, CP01, you can see staining of tonsil using CD8. Those are the small red cells over there to the right. Pan-CK expression alone is shown in yellow, and PD-L1 expression alone is shown in blue. And you can see a clump of cells there over about 11 o'clock, and some also blue cells alone in the center of the section. Where PD-L1 and pan-CK are co-expressed. The yellow and the blue chromogens produce a green color. So by carefully selecting our biomarkers and our chromogens, we can actually look at co-expression of biomarkers by chromogenic staining. Now, we're used to seeing this on fluorescence, but it's unusual to be able to see it on chromogenic staining. By the choice of these chromogens, we were able to detect it using ultraplex. The middle panel, CPO2, is looking at CD4 and CD8 cell infiltration into tumor nests. So this is colorectal cancer. You can see it with the yellow staining, and you can quite clearly see infiltration of blue CD4 cells and red CD8 cells into the tumor nest there. The panel over to the right is CPO3, so the CD68 cells are shown in red. The CD163 cells are shown in yellow, and where there's co-expression, they appear as orange. You can see a lot of cells which are PD-L1 expressing alone. Those would be the tumor cells. And you can also see some blue PD-L1 expression on the kind of orangey macrophage cells, but it's much harder to see when you're looking at 3 biomarker expression by chromogenic. Really looking at double biomarker expression would be the sweet spot. On this slide, I just wanted to showcase how we can easily adapt. Here's an example. This is CPO1. So instead of staining for pan-CK in yellow, we're staining with CD68. If you look at the farmost panel on the right, this is lung adenocarcinoma at the highest magnification. The small red cells are CD8 cells, which are mainly there in the stroma. The blue cells alone will be the tumor cells, stained with PD-L1. And then the CD68 cells you can see as yellow cells. Now, quite a few of those also have expression of CD8 shown in red, and this alarmed me, I have to admit, when I first saw it, but when we showed it to our pathologist, he said that that was quite normal in the lung biopsies, that you might see CD8 on your CD68 macrophages. And if you look about 12 o'clock, you can see a couple of cells there which also show membrane staining with PD-L1. So in this section, you see both tumor cells and macrophages expressing PD-L1. In this next slide, it's the same panel, but we've replaced CD68 with CD163. Again, on the far right, you can see the CD8 killer T cells. The CD163 M2 macrophages are shown as the yellow macrophages with some of them have red on them. But I will be honest, in this particular section, I can't see any co-expression with PD-L1, but it doesn't mean to say that there wasn't any in this section, just not in this particular view. And again, I wanted to show you the CD8 PD-L1 and pan-CK panel, because this is a very nice illustration of the co-expression here. The CD8 cells in red are shown again mainly in the stroma. The yellow cells are the tumor cells. And then the areas where PD-L1 is co-expressed on the tumor are shown as that very strong green color, which is really interesting that it is very concentrated on the interface with the stroma, the outward facing interface, whereas the center of the tumor has very little PD-L1. So I'm always fascinated by these images. Here on this section, we're showing very different patient profiles. This was a non-small cell lung cancer TMA, so 3 different patient samples using the CPO1 panel. The panel over there on the far left shows pretty much all of the tumor cells also co-express PD-L1. There are some red cells there in the CD8 cells in the stroma, but I don't see any of them infiltrating into the tumor. In the middle panel, you see a lot of CD8 killer cells in the stroma. A few of them are infiltrating into the tumor, but the tumor is very noticeable in that it expresses no PD-L1. Over there on the right, you can see quite a few CD8 cells infiltrating the tumor, but the tumor is varied in its level of PD-L1 expression. You can see some cells which express it, but also some tumor cells which don't. So again, 3 very different profiles, all from the same tumor type but different patients. On this slide on the left, I'm just showing you a higher power view of the slide I showed you earlier so that you can very clearly see the membrane staining for HER2 in blue, the red nuclear staining for the estrogen receptor, and the orange nuclear staining where you get nuclear co-expression of estrogen receptor and progesterone receptor. Over there on the right is showing staining of tonsil, and it's just showcasing a 4-plex chromogenic panel. In the center of the slide, you can see red staining for CD8 killer cells. Over there on the left, you have pan-CK staining alone. And the green area about 7 o'clock is where you get co-expression of pan-CK and PD-L1, which is green. If you look there over to the right, that would be a germinal center showing rapidly staining cells, which are detected using an anti-Ki67 antibody and the LICA green chromogen. So these two greens are quite different. And I have to say, I really like the LICA green chromogen. It shows very, very nice staining. This is just another 4-plex panel. In this case, what we've added in to CPO2 is staining of macrophages with a black chromogen. Over there, staining tonsil on the left and lung adenocarcinoma on the right. So on this slide, we show the reproducibility of the chromogenic staining on lung cancer biopsies. So we took 10 slides and we stained them with the CPO1 panel. In these slides, we're using the purple hematoxylin. And I wanted to mention that because all the previous slides had used the pale blue hematoxylin, just so that you could see the difference between them. If you look at the sections themselves and also the bar graphs to the right, you can see that there is very strong reproducibility slide to slide. The error bars are very small, staining both for pan-CK and for the pan-CK PD-L1 co-expression. You can see most of the pan-CK expressing tumor cells also express PD-L1, both from bar graphs and also if you look at the slides, the co-expression is shown as that dark green brown staining. Also the CD8 T cells in red, again, very reproducible slide to slide. In this slide, we're showing image analysis of breast cancer stained with HER2, ER, and PR. We use multiple different image analysis programs, and we are image analysis agnostic, but in this particular analysis, we were using Visiopharm. The image over there to the left is the actual of the breast cancer sample. The image in the middle is where we applied the mask, which is the thresholding. And then the image over there to the right is with the mask added, where we are showing, for example, HER2 staining alone. The cells are shown in green. And on this particular slide, we had about 75,000 cells staining HER2 alone. The estrogen receptor staining alone is shown in blue, and there are about 34,000 cells. And then the estrogen receptor, progesterone receptor co-expression is shown in red, and there are about 33,000 cells in this particular slide. This is image analysis of lung cancer, again using Visiopharm analysis. The panel staining it is the CPO1, CD8, pan-CK, and PD-L1 panel. The two images over there to the left, where we analyze specifically the stroma areas, and the top with the mask off and the bottom with the mask on. So the red cells are the CD8 staining cells alone, and the blue cells are the cells which express PD-L1 alone. The two panels there over to the right, we analyze specifically the tumor cells. The PD-L1 pan-CK positive cells are shown there in the bottom right panel as blue cells, and then the pan-CK tumor cells which express pan-CK alone are shown in brown. And there we've analyzed, and you can see in the bar graph, we've analyzed about 350,000 cells, and it shows the percentages of those different cell populations. So the Ultraplex kits are available for staining 10 slides, and they also include an additional aliquot of secondary antibodies alone, so you can add a negative control, which is stained with the secondary alone. It requires the use of the bond-direct staining system buffers, reagents, and detection kits. And of course, we have larger kits available on request. So in summary, Ultraplex Chromogenic Multiplex IHC is a rapid plug-and-play technology for tissue profiling. It allows automated detection of 3 markers on an autostainer in 4.5 hours, using primary antibodies from any species while employing a single antigen retrieval step. The panels I've shown you and all the ones that we offer are FFPE tissue optimized for key biomarkers. This allows rapid scanning and avoids autofluorescence issues. So I'm a big fan of ultraplex fluorescence staining, but there are some tissues, for example, like prostate or lung cancer, which sometimes have autofluorescence using a fluorescent readout, which obviously you avoid using the chromogenic readout and are able to very rapidly scan your slides. And we also have custom panels available. We get chromogenic detection of cell subtypes co-expressing biomarkers using our optimized chromogen combinations. So I want to change it up a little bit here and talk about fluorescence staining. So using Ultraplex Fluorescent Multiplex IHC, and this is a simple, flexible, fast, and fully automated technology. It generates beautiful images and data on biomarker expression and spatial localization of key cell subtypes using the bond-direct staining system and the Aperio versus scanner. The flow diagram, again, is the one that I've shown you previously, but here I just wanted to emphasize in the panel below the options that you have for fluorescent staining. So we have ready-to-use panels for fluorescent human multiplex. We have modular select your own panels for both human and mouse, and we have custom panels available again for human and mouse. You can run these panels using the bond-direct staining system, the 7.0 staining software. The fluorescent staining protocol will be added to the next protocol upgrade, which should happen very soon. But in the meantime, all of the CellIDX suggested protocols are available on the CellIDX website, so you can download them all. The image over there is produced using the Aperio versus scanner, and it showcases very nicely the staining of the tumor in red. The orange area is a co-expression of pan-CK with PD-L1, so that's tumor cells expressing PD-L1. The green infiltrating cells are CD8, and then I think you can see some macrophages which are outside the tumor. Those are the purple expressing cells. Here I'm showing you a higher power of the fluorescent multiplex, the breast cancer panel that I showed you earlier. And again, you can see the red membrane staining for HER2, the blue and green nucleus staining for estrogen receptor and the progesterone receptor, respectively, and the turquoise staining where they co-express, and then the staining for Ki67 in the nucleus is the pink stain. So again, it's a simple 2-step staining procedure, which is very rapid. It's easy to implement. It has a single antigen retrieval, which is rapid and tissue conserving. It's antibody species independent. It uses cocktails of antibody, which allows simultaneous detection, and the panels are all FFPE tissue optimized. So routinely we produce 4-plex panels because those are they can easily be detected on most main scanners, and certainly could be detected on the LIGO versus scanner. But we can also produce 5-plex panels. But what we often do with our 4-plex panels is serial section overlay. So for example, over there at the upper left, you can see the breast cancer panel I showed you earlier. And at the bottom right, you can see a basic immune panel. And even without overlaying those 2 panels, you can see in this patient, they have quite a strong immune response, but the problem is that it's not gaining access into the tumor. And I wanted to just pause there and also emphasize a little bit about the speed of the rapidity of these sections. We talked about a few minutes ago about how rapid it is to do staining, but I also wanted to emphasize here how rapid it is to rearrange those panels. So for us, if we want to rearrange a 4-plex panel, say to add in an additional 1 or 2 biomarkers, all we need to do is remove those antibodies from the cocktail and add in 1 or 2 antibodies into the cocktail. And we can create those very rapidly. So it helps you to really rapidly evolve your research. You might stain one section and then scan it and analyze it in one day. You can look at the data. It might give you an idea of a different stain that you wanted to do. You could order another modular panel. It will arrive very rapidly. You can even stain the serial section and then overlay them, as I've shown here, to really enable you to ask questions and address them and evolve your research very, very rapidly. So that is one of the other advantages of Ultraplex. Over here, I'm showing a very basic T-cell phenotyping panel using CD4, CD8, FOXP3, and TBET. If you look at the 4 small images going from left to right, you have membrane staining for CD4 and nuclear staining of FOXP3, so that's a Treg. Next to it, you have membrane staining of CD4 and nuclear TBET, which is a Th1. Next to that would be membrane staining for CD8, nuclear TBET, which would be a Tc1. And then right next to that, we have membrane staining for CD4, and then nuclear expression of both FOXP3 and TBET. And you can see just a couple of those cells in the larger image. So that's a very rare subpopulation, which may be a suppressor cell for Th1 cells. But in this particular patient's, melanoma patient's tissue sample, you see quite a lot of the Tc1 cells. Just wanted to insert this slide here because we have expanded our Versa scanner to actually detect 4 different biomarkers plus DAPI using a Megastokes dye and a custom filter cube from Chroma. So if you look at the table of fluors, the CL490, 550, 650, and 750 are the fluor 4 set we usually use with our pre-made panels. And those are regular fluors using the usual 25 nanometer Stokes shift. If you look at the lowest line, the 5th one down, that's a Megastokes dye. So that has a much greater Stokes shift. It's about 130 nanometers. So by using this dye with the extra filter cube, we can actually detect 5 different biomarkers. As long as the expression is strong, you're able to do this. So it gives you an additional flexibility. So here are our ready-to-use panels. So they're 4-plex panels, and if you work in the area of immuno-oncology, they'll make perfect sense to you. We had a demand for these, but a lot of people will be asking, say, for example, they wanted to biomarker for another biomarker, and we have a lot of different qualified antibodies, and we're always developing new targets. So what we did was we released what's called Ultraplex Modular. So this is really selecting your own multiplex panel. You look at the menu of qualified biomarkers and you get to choose the 4 that you want to include into your panel. You choose your fluor 4 combination and then you select hits via the 10 or 50 slides. And we can also obviously make custom panels on demand. And this again is just showing the CD4, CD8, FOXP3 TBET panel. Now I wanted to change to show you Ultraplex mouse modular panels. So again, this is the same approach that we had for the human. All the antibodies are FFP tissue optimized. You get to choose the 4 biomarkers you want from our qualified panel. Again, choose the fluor combination and select kits for 10 or 50 slides. And again, here we can also make custom panels on demand. This is just showcasing different modular panels that we've created just to give you an idea of what we can produce. If you look over there on the left panel for the FFPE spleen, this is a CD3, CD4, CD8 FOXP3 staining panel. FOXP3 is in yellow, so you can see a lot of nice yellow nuclear staining, got a lot of Tregs in that staining of the spleen. If you look there on the right, you can see staining of a mouse lung cancer model, KRAS tumor. There are very few yellow nuclear staining cells. There's very few FOXP3 in this particular section. We show green CD8 cells, so there's quite a few CD8 cells there and quite a lot of purple CD4 cells in this particular area of the section. This panel stains for CD8, FOXP3, F480 is a macrophage marker, and CD11B. If you look at the KRAS tumor stain there on the right, there are a lot of F480 macrophages there, and very, very few FOXP3 yellow cells, and just a few CD8 T cells. So this is an interesting panel. Over there on the right, you can see a lot of B cells staining in green, stained with B220. You can see a few red macrophages, and you can see CD11B cells in purple. That would stain a variety of different cells, but we've also stained for myeloperoxidase in yellow, so where you get double expression of myeloperoxidase and CD11B, which would be neutrophils. You can see these very bright purple and white cells. Again, over here on the right, these are staining with a BondRx and scanning with the Aperio versus scanner. This slide shows staining of different mouse and genetic tumor models. So the left is a breast cancer model, and what's really striking with this staining is that, so the panel is CD8, CD163, FOXP3, and Ka67. There are a very large number of proliferating CD8 cells in this particular view of this breast cancer model. So those have membrane staining in yellow for CD8 and nuclear staining in purple for Ki67, and then a few green M2 macrophages. The pancreatic tumor there on the right, there's a lot of proliferating cells, but it looks like those are mainly tumor cells. You have a few green CD163 cells, and then quite a few yellow CD8 cells and a few FOXP3 Tregs. Over there on the left, you have the CT26 colon tumor model. Again, a lot of green CD163 macrophages and a few yellow and purple rapidly proliferating T cells. You can see a little nest over there about 4 o'clock. The tumor to the right is the hepatic tumor model. You can see a few CD163 cells and a few CD8 T cells in yellow, which don't appear to be proliferating. Now, in none of these are we saying whether these are hot or cold tumor models, but these are just areas where we focused, where we saw staining. And here I just wanted to show you an example of mouse chromogenic multiplex IHC. So these are panels on request. This is showing normal mouse colon stained with pan-CK in yellow. And then close to that, you can see the CD163 macrophages stained in red. And lower down in the image, you can see a lot of blue CD8 T cells. And finally, I wanted to talk to you a little about the Ultratag multiplex labeling kits, which allow you to develop your own multiplex IHC panels. So this is a very simple labeling procedure for 50 micrograms of antibody. Your antibody needs to be a high affinity antibody, and the antibody needs to be about around a mg per ml concentration without additional BSA. So the tagging is obviously antibody species independent. It's very efficient tagging, and it takes about 20 minutes hands-on time, which will be about 2 hours start to finish. So we have Ultratag kits available to make up to 4-plex fluorescent panels and 3-plex chromogenic panels. So if you look at the list of kits, you need 1 kit to label each antibody. So if you wanted to make a 4-plex, which you could use on the say for example, the Versa, you would probably choose the Ultratag 490, 550, 650, and 750 kits. If you could do spectral and mixing, you might choose the 490, 550, 595, and 650 kits. If you wanted to do chromogenic make a chromogenic panel, you would choose the AP, the HRP1, and the HRP2 kits. So these are inclusive kits which contain the tag labeling reagent to label one antibody, and then they also include either the fluorescent labeled or the enzyme labeled detector antibody. And I think that we've come to the end of the talk, but I would be delighted to take questions if anybody has them.
Thank you, Helen, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. We have some great questions coming in from our audience. We'll start with this one. Helen, what are the differences between the set panels, modular panels, and custom panels?
Thank you, Kaylee. So the set panels are the human fluorescent panels and the human chromogenic panels. So those would be the the HI1 through HI6, which are the fluorescent panels, and they make a lot of sense. That, say, for example, HI4 stains CD8, CD68, pan-CK, and PD-L1. So that would be looking, if you're working in immuno-oncology, it would be looking at infiltrating CD8 cells and PD-L1 expression on either tumor cells or on macrophages. The CPO1 through 3 are the chromogenic panels. So those will be all of the pre-made panels. So if the panel that you're interested in is there, that's great. If it isn't, then you can look at our modular panels. So those would be both human and mouse fluorescent multiplexing. So there you basically look at the menu of qualified biomarkers. You choose the 4 that you're interested in and the fluorophore sets, and just place your order. And the ready-to-use panels, obviously they're off the shelf, they're shipped immediately. The modular are available very, very rapidly. It might be a couple of days. If we have to rearrange things, it might take a little bit longer, but it's very, very rapid. Now, if you can't find the panels that you want, either with the pre-made panels or with modular, then you can order custom kits. So any of the chromogenic panels, which we don't have available pre-made, you could order those as a custom panel. Or if you have antibodies that you particularly want to use, maybe they're up against biomarkers that we don't already have, we can make those in chromogenic or fluorescent form as custom panels. So those are the 3 options. The custom obviously will take a little bit longer, but we're also very efficient at that too.
Great, thank you. Another question I have for you here. What fluor sets are available for the human and mouse fluorescent panels?
So the human fluorescent panels use our A-panel fluors. So that would be CL 490, 550, 650, and 750. So those are the ones that we routinely use on our Leica Versa. If you have a scanner that can spectrally unmix, you might want our B-panel fluors, which are 490, 550, 595, and 650. We also have other fluors available. So for example, CL 532 is very popular, particularly if people are interested in using the nanostring geomics platform technology. Often they will order a panel using 490, 532, and 650. So there's a lot of flexibility there with the modular panels.
Great. Now, Helen, can this ultraplex technology be used with xenograft models?
Thank you. No, that's a good question. We have not done the staining in-house, but theory it should be perfect for looking at xenograft models. So sometimes people want to use mouse monoclonals to detect the human tissue grafted into the mouse host, and then use an anti-mouse species specific detection reagent, where sometimes they might see cross-reactivity on the mouse tissue. So I think, as I've said before, because we tag our antibodies, we're not detecting them by the species, we're detecting them by the tag. So it should, in theory, be a perfect solution and avoid any mouse-on-mouse problems you might have using mouse monoclonals in the human xenograft, patient-derived xenograft models.
Great. Another question I have for you here. You showed staining using red, blue, and yellow chromogens. Can you use others?
Yes, actually, I also showed the images using the black chromogen and then also the Leica green chromogen. So I like the Leica green very much. We actually have a red, blue, green protocol on the BondRx. I don't think it's available for most use, but it is programmed in there. So if you call Leica, they might enable you to use the red, blue, green chromogen protocol too. If you wanted to use other chromogens, I would just suggest that you give CellIDX a call. We've really optimized it with the red, blue, yellow in terms of the order and the biomarker chromogen combinations. But if you wanted to try something else, just give us a call and we can chat about how feasible that might be.
Perfect. Next question here asks, you said you supply both blue and purple counterstains with the kit. Why would you choose one versus the other?
Well, there are two answers to that question. The first would be cosmetic. You might just prefer one to the other. I showed you in the serial section staining with the light purple counterstain, and actually I think you can still see the image of the pale blue counterstain on the screen. So you might just prefer one to the other. The second, probably more important answer is in terms of analysis. So depending on which analysis program you're using, you might find that it works better using the pale blue or the pale purple hematoxylin. So we include both so that you can try it ahead of time and choose the one which gives you the best definition and the best analysis.
We have time for a few more questions here. We'll proceed with this next one. Do any of your panels work on non-human primates or other species?
That's a really good question too. I did look quite a while ago to see if there was cross-reactivity, and I did not find very much data on that. Not to say that they don't, just that there's a lot of data at the time. But we're very transparent about what clones of antibodies we use. So they're on the website, so you can easily see what we're using for the staining. But I think that the best solution for that, whether you're staining non-human primates or maybe even dog tissue samples or cat, would be the Ultratag kits. So as long as you've got a good high affinity antibody at about a mg per mil with no BSA, the Ultratag kits should work very well. And when I say a good high affinity antibody, what I'm really talking about is an antibody that titrates down well. So when we qualify antibodies, one of our first steps is that we do a titration from 1 microgram per mil to 0.2 to 0.04 micrograms per mil. So a good antibody will stain well at below a microgram per mil. So if you have that kind of antibody, it would be a good candidate for tagging with Ultratag, and then you could tag all your own antibodies, and regardless of the host tissue, you can, if you have the antibody, you can create an ultraplex panel, either fluorescent or chromogenic.
Great, thank you. Next question, where can I find the protocols to run the BondRx?
Well, if you have the version 7.0 software, the chromogenic will be pre-programmed in there. And I believe if you have 6.0, you can always upgrade it to 7.0. For the fluorescence, they're going to be on the pre-programmed protocol very soon. But in the meantime, if you go to the CellIDX website, and I think it's the support menu at the top, if you pull that down to the protocols tab, then you can download any of our protocols, and that would include the fluorescent staining protocol that we recommend for using on the BondRx.
Great. Well, it looks like we have time for one more question, so we'll wrap up after this one. Can I use any imager or scanner for my stained slides?
Well, for the chromogenic slides, it's just using bright field microscopy, so you can use any bright field microscope or scanner. For the fluorescent stain slides, just make sure that you use the panel, the fluorophore panel that works with your scanner. Like I said before, a lot of people have scanners which could detect the A panel fluor, so that's 490, 550, 650, and 750. Other people can do spectral mixing, so they would want the B panel fluor. So just make sure that you choose the panel that you can actually see when you scan afterwards.
Great. Well, thank you again, Helen, for your time today and your important research. We would also like to thank Labroots and our sponsor, Leica Biosystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.